I am at Western University, and I will switch a little bit now away from Mycoplasma, because here in Canada we are scared of working with the synthetic cell. Actually, some of the reasons are that the biosafety level are a little bit higher, and one of the species that I was interested for a while is Acholeplasma. So I'm gonna tell you about this work. Now, some of the research in my lab, or at least this is just starting. We are starting, but one of the major topic is, as you heard a little bit from Miriam last week, that we want to convert some free-living bacteria into organelles. And there are a number of options here to choose. We started with few different ones. So since we cannot work with the synthetic cell yet at the moment, we selected few different species. So there is Acholeplasma. So for molecules, or what we call custom bacteria, we are trying to develop this for Acholeplasma laidlawii, Haemophilus influenza, and we did a little bit work just with Mesoplasma florum. We also would like to convert Rhizobia as nitrogen-fixing organelle. So we started developing tools for 3 different species. So essentially, in order to enable all this thing, it's always you need extra genetic tools to enable any kind of manipulation. So for Acholeplasma was, at least in my hands, one of the hardest organisms to ever work with. When I was still working at the Venter Institute, essentially I was unable to really transform it. It was very easy to develop spontaneous mutation that were resistant to different antibiotics. So that research kind of suffered. But fortunately, I had I have a really great student that took on this challenge here when I'm in Canada, and decided to actually go ahead and try to change that. So we're hoping that once we have improved genetic toolbox, we're gonna have better transformation, such as better transformation methods, additional plasmid. We can have better ways to clone genome in yeast and also develop genome transplantation. We can attempt to create those synthetic organelles, starting leaving them obviously in yeast. And then could we also use this technology for creating synthetic cells, as we are doing right now with the Mycoplasma, and potentially make some hybrid cells. So here is summary of some. It's probably not completed, but the question is, could we use some of the existing tools that were already developed for the Mycoplasma, that we could create, essentially synthetic cell of Acholeplasma, in the existing system. So I listed here whole bacterial genomes that were cloning yeast, and you have Haemophilus influenza, some algae, and there's a lot of Mycoplasma, including Acholeplasma. Now, for transplantation, unfortunately, was never obtained for Acholeplasma. And if you look at the transplanted genomes, most of them use Mycoplasma capricolum as a recipient, and some of them were booted up to put it up in their own cells. Now, what is one thing that is shared so far, because between all the transplanted genome, is that they use this non-standard genetic code with UG8 code for tryptophan instead of stop codon. So Acholeplasma is different in that sense, as it uses standard genetic code. So that's another potential reason that we could not transplant it in for example, like what is right now kind of the most universal recipient, Mycoplasma capricolum. Also, what is interesting, and we are not really doing much work here, but we're looking for collaboration here. Acholeplasma. So once we're able to have synthetic Acholeplasma and have genome transplantation working, we potentially can start creating hybrids with Phytoplasma, which cause disease in plants, but Phytoplasma really cannot be easily propagated in laboratory conditions. So they are very closely together related, Acholeplasma and Phytoplasma. So hopefully that will be a good resource for that. All right, so we just submitted last week a little paper. It is, you can check it at bioRxiv right now, where when we develop what I kind of almost call it like a tipping point to enable the research with Acholeplasma, to enable the research of creating synthetic cells and make protocols a little bit more streamlined. So what we did, we are proposing two different strains as a donor and recipients for the genome transplantation. So that's what we have right now. The genetic tools are or were developed a little bit for some of the species, but they are still limited. One of the genome was cloned in yeast, but the genome was never assembled from synthetic fragments in yeast. Genome transplantation from bacteria to bacteria was never performed, and definitely not from yeast to bacteria. So those are the missing puzzles, essentially, for us to kind of solve. And we would love to solve it in this coming year, hopefully all that. Okay, but so what do we have so far? So the two strains that we are proposing is to use Acholeplasma laidlawii PG8A as a donor strain, and strain 8195 as a recipient. And they have very similar sequence identity to the pair that was used for Mycoplasma, Mycoides, and Capricolum. So they are a 93% sequence identity. But what is the reasons we selected them, one donor and the recipient, is that the PG8 has a lot of restriction systems, and the 81 doesn't, or at least we identify one so far that has mutation in it. So that should be restriction minus and also recombination minus strain. They are both around 1.5 megabase pairs and GC about 32%. Now, as we start doing the genome, potential transplantation, we want to be able to have quick visual screens. So one thing that we notice is that they both produce carotenoids. So important thing to add is that you don't need to add cholesterol to the media. So media is cheaper, so it's a little bit easier to work with it comparing to the synthetic cell in that terms. And so when they produce those carotenoids, one of the strains is a little bit more, has more pigment, looks more yellow than the other one. So that could be visual screen. And we also have a quick multiplex screen. So we have a number of primers distributed throughout the genome that when you do the PCR and use them on both strains, the PGA specific primers will amplify on one and not the other one, and vice versa. So those could be easily distinguished as we move forward. Now, this is past research. So I just want to show you this because this was done it was published 10 years ago. So the first genome was first cloning of the whole genome was published in the ACS 10 years ago, but the research was kind of a little bit slow. And even with the cloning, Acholeplasma created a lot of problems because the whole genome initially couldn't be cloned. Small piece was never clonable. And then we found out that essentially by narrowing down the pieces to less and less, we found out that there was one fragment. There was essentially on one fragment, there were a couple genes, and one of them was this extracellular endonuclease that if you had it, this gene was able to be expressed in yeast and be toxic to yeast. So again, having the standard genetic code and cloning things in yeast may be problematic because of the toxicity. But by inactivating that gene essentially and putting the plasmid, the yeast artificial chromosomes right into the gene, we were able to clone the whole gene. And that was actually published in the very first issue of SynBio. So 10 years passed and not that much happened with engineering of Acholeplasma. So towards that goal, we said, well, we need to, even there are some methods for transforming by electroporation and PEG method, polyethylene glycol, they were not working that great in our hands still. So Daniel Nusifora, who is a PhD student in my lab, he started checking other things. So one of the first things he said, well, let's check more of if there is more of extracellular nuclease in those species. So essentially, he took some live cells and mixed them with plasmid and incubated at 34 degrees for 0 or 30, 60, 90 minutes. And you see at 0, when they're still sitting essentially on ice, the plasmid is there, and after 30, the plasmid is completely gone. We should check obviously the how long the earlier time points, but it's completely gone. If you add EDTA, you can inhibit that, and here is just a plasmid without the cells. So this is in the strain PGA8, the one that has restriction systems and potential nucleases. Now in the other one that we are proposing as a recipient, you can see that they were a little bit they were not degrading as much. When you look between the 0 and 30 minutes, there are still some plasmids left, but they do disappear. So it is a better strain, definitely has less of this activities, nuclease activities, but still has some.
Bogumil,
So,
are you doing this in the presence of calcium?
This one was actually, Daniel, what's the calcium there? I don't think so. That was just in the sucrose buffer, as far as I know.
Okay.
And Daniel is here on the call, but yeah. So maybe I will just kind of revisit the end. So that was, but essentially learning that we can add a little bit of DTA and that would inhibit that. We optimize electroporation protocol. So, and I'm not gonna go through this because it's kind of boring, obviously, but now we can just take 1 microliter of the culture in exponential, pre-treat it with EDTA for 10 minutes and do a couple more washes after, and we're getting thousands of colonies. So that was actually great. So that is, and I know when I was at Venture Institute, I've never got a single colony. So for me, that was a little breakthrough. Now we are using here this this strain that's supposed to be restriction minus, but again, still has some nucleases. So the protocol is there. You can look at this on bioRxiv. What we also did next, we developed plasmid that has this Acholeplasma origin of replication. So essentially, it's the DNA A with some DNA boxes around it. So we created this plasmid called PAL1. It has also yeast elements, E. coli elements that can replicate. So that's the 10 kb plasmid. It has tetracycline gene. We put tetracycline gene and puromycin gene. The puromycin didn't work, but the TET worked very nicely. We could select it, and essentially we learn, how to grow them. So when we pick up colonies after 4 to 6 days, they are good. They are always good. When you go past 10 days, you may get spontaneous mutant. So it's we can clearly distinguish that. We also use this published plasmid, PNZ18, which has neomycin marker, which is half the size and also work very well. We use it 200 for neomycin, and if you use that selection, you don't have essentially background for at least 12 days. Those plasmids don't integrate when they propagate it. They can be recovered. So that's what you see at the bottom of the gel. We recover them in E. coli, and when perform diagnostic digest on both of them, here are 5 colonies, and they stay without, so they were not incorporating. Now, if you look at the PAL1, we actually wanted to test, is this plasmid, if it cannot be replicated with the origin, Acholeplasma origin, can it incorporate? So we added 2 homology regions, homology 1 and 2, about 1 kb, and this would be flanking the toxic gene. So we said, well, potentially could we incorporate some of that plasmid there, or could incorporate it even at one location? But it doesn't seem that it does. So again, it proves that it's a combination minus, most likely, or there was at least no pressure to incorporate it. And then, so another thing is that that is going to be potentially good recipient. One more thing to create. So now we have a really good method to move plasmids. So not a problem. We can load pathways, for other genes and start developing all the different tools. So electroporation or transformation of Acholeplasma shouldn't be problematic in anyone's hands, and it's very simple protocol. It's simplified and use very little culture. The next thing was, could we use transposon? Because transposon, we all love transposons, TN5, for finding if we can find the minimal set of genes, we can add different functions, and it works. The protocol is not as reproducible as the plasmid, so we can get anywhere from couple colonies to couple hundred. So we are still optimizing why the transposon is still so finicky a little bit, but we can get already hundreds of colonies. So we can pull them and start performing some other analysis of minimal genome. But we also, by doing that, we essentially added transposon, like again, TET cassette. This one is to the PGA8 strain that I was never able to transform in the past myself. And now we have a strain with that carries the transposon, and they can grow on tetracycline 5. So it is good donor strain, at least between Acholeplasma to Acholeplasma transplantation. And we can confirm it by multiplex, or by just PCR that it's there, and by growing them on selective plates. So So essentially, that seems like just a little bit a start, but I think that for me that is completely gonna change the things, because now we can test a lot of those different things, even some of those things that were recently today presented, putting more landing pads, developing other methods. So anyone that is interested in any aspect of this research, we would like to have a meeting sometimes in the next couple of months. We want to share our results as quickly as we get anything, and hopefully some of you would have uses for our strains. And again, towards transplanting of genomes with standard genetic code, this could be one of the easier one now, because it has very similar properties to Mycoplasma in terms of the cell wall. It's still relatively small, 1.5 megabases, and then we can clone those things in ease. So anyway, someone please email me, and we're gonna go from there. So I want to thank you, everyone in my lab, especially Daniel Lucifora, who is the PhD student leading that. We had, and she's still in our lab, Nidhi, who is working on, who's undergrad student, then who recently graduated, helped with all the sequencing, and then Kevin and James for providing the 8195 strain. And we also got the plasmid from Dr. Hamon at Amsterdam, and we are talking to Carol our possibilities, and hopefully this will just kind of continue improve. So thank you, everyone, and I will stop here, and I guess stop the share screen.
Very nice. Questions? Bogumil, have you thought about, so Dan Gibson was approached about the idea of looking at mycoplasmas as models for the citrus greening bacteria. I forget the name of the organism. But
are they phytoplasmas? Those would be phytoplasmas, right?
No, it's not. It's a conventional bacterium, but Librobacteria or something. Anyway, this could be, but phytoplasma, I mean, let's face it, Acholeplasma is not that much of a problem. It causes some sheep issues, but the phytoplasmas could be a huge deal if you could engineer them to not be pathogenic and supplant phytoplasma infections in commercial, I mean, in economically valuable plants.
Yeah, so I mean, that is one of the goals that, Acholeplasma is great to start because we can work in the lab with it, and then so the question is like how much we can install it right now in Acholeplasma. So now having those plasmids, multiple plasmids, we can start installing them on the plasmids, hopefully in the genome, but then later we could be potentially making some hybrid genomes, right? So and so I know we are interested here, and to talk to Carol a little bit. So if we have these tools, we're gonna start helping and again creating this, and Acholeplasma would be just again another, as we're talking about couple universal recipient cells. So Acholeplasma, we're not gonna have one universal, but we may have multiple ones, and for certain family, Acholeplasma could be the one. To booting up other organisms. So going from again, not standard genetic code and couple other properties, easier like cheaper media. The biggest problem for me is still that it grows a little bit slow. We're getting colonies actually with the neomycin selection. We're getting colonies within 3 days. They grow almost faster with the antibiotic. It's amazing. 3 days, and TED we more like 4 to 6 days. Can we find a better Acholeplasma in the future that is going to be growing even faster? But so it's just the beginning. We're hoping that again with the collaboration, we're going to have a lot of data to show next year at this meeting.
You probably know this, but the late Swedish mycoplasmologist Aki Wieslander used Acholeplasma as a model organism to investigate membrane biology in a day when his real interest was using this as a model system for what goes on in eukaryotes. And he later on moved to eukaryotes, but you might look at some of his old papers and see if there are issues that are still unresolved that you might be able to address. Other mycoplasmologists who are, on the, present here might chime in on this. I have not looked at those papers in 15 years, but it's something that you might look into. Kim or Pascal, Carol, you got any thoughts about this?
Yeah. No. Okay, just maybe we'll talk about it later. But here we were also interested in developing genome transplantation with Acholeplasma, but frankly, so far we are a little bit slow about the genome transplantation development. However, we're trying to, on the other side, in fact, we're trying to clone the phytoplasma genome in yeast. It's a hard task because, as you know, we cannot grow them in vitro. So, in fact, we had to work with either infected plants or infected insects. So it's what we've done. And so far, we could isolated the genome in agarose plugs, and it looks great on PFGE. And we could also clone a part of the genome, but not the whole genome so far. But we will continue, and we have someone in the lab working on that aspect with us. So maybe at one point we can join the two projects if in Bogumil Karas lab, you have the transplantation working, and here we have the cloned the phytoplasma genome. Maybe we can join the effort and our efforts.
Yeah, absolutely. I will, I always say this, but thanks to Carol, I am in synthetic biology. I was so impressed with the genome transplantation that I said I want to do this. So that is like working with my idol. So absolutely, I'll be like so happy. And then actually, if we cannot clone it directly from the strain, could we, with the power of SynBio, could we just synthesize the genome if we have a good sequence?
Yes, we can do this.
Maybe one technical difficulty will be in this hypothesis. Phytoplasma genomes are even worse than mycoplasma genomes because they have many repeats, and in some parts of the genomes, the GC content is really, really near zero. So in terms of synthesis, I guess there could be some problems.
Right. So propagating in E. coli or other, they would have to probably be done right away into yeast, synthesize and assemble in yeast because they're going to fall apart in E. coli easily, right? The low GC are really hard to, or impossible almost to have in.
I wouldn't talk yourself out of trying it.
Yeah, for sure. Yeah.
Thank you, Bogumil. So, and I'm going to talk about planned efforts to try to expand transplantation into conventional bacteria as the last talk of the day.